Photosynthesis, a Callian show.
This is the chloroplast.
That is the outer membrane.
This is the inner membrane of the chloroplast.
And that is a granum. It's a stack. It's your breakfast. It's the entire whole thing.
Of thylakoids. Now this is a singular thylakoid. It is basically the pancake of your breakfast.
It's lovely looking, isn't it? It's where all the big things happen. It's great.
In the thylakoid membrane, you will find the light reaction,
where light goes in and stuff happens. Outside of the light reactions, the dark reactions happen in the stroma, in the fluid around the thylakoids.
Yay! The light reaction.
And now we go to the thylakoid membrane, where stuff happens. Okay, so what we're looking at right here, the bigger picture is a magnified vision of the thylakoid membrane, which is a small segment of what I am drawing right now. So that's what that is. The first thing there is PS2, Photosystem 2.
Photosystem 2 comes before Photosystem 1 because the people who discovered this hate you, and they want you to not be able to remember it. Okay, now this is Photosystem 1. Photosystem 1 happens after Photosystem 2. We will get to the details soon. And then this is the ATP synthase.
And we're going to talk about Photosystem 2 first, though. So, and apparently ATP synthase looks like a jellyfish. A jellyfish. Photosystem 2.
In the beginning of Photosystem 2, sunlight enters the photosystem from the sun. It comes from the sun.
And breaks water, H2O, into H plus ions and oxygen. And it gives off electrons into this giant proteiny thingy here. And that electron gets excited by the sunlight.
And it goes and it freaks out. It's so excited. It has so much energy that it goes up here. And there are these little receptor things that receive the electron. And basically it gets turned into chemical energy is kind of what's happening in this photosystem.
It gets super excited, and then it goes into this little carrier molecule here, which takes that super duper excited electron that's been sitting on chlorophyll, which functions at 680 nanometer wavelength red light.
So basically the reason chlorophyll is green is because it reflects green light. That's what it means that it's 680 nanometer, is that it reflects green light.
And it absorbs red light, which is used to turn electrons into chemical energy, basically. And that super excited light-filled electron gets stuck onto this carrier, which we mentioned, but this is out of sync, but that's okay. So that goes onto that little carrier, and it goes off, and it goes off to do work, because it's super energized and excited, and so it can do things.
It leaves Photosystem 2, and what happens next? So what we're looking at right here, this is in between Photosystem 2 and Photosystem 1. So it leaves Photosystem 2, and then it
goes to this little protein pump. This is a protein embedded in the thylakoid membrane where this energized electron is going to power hydrogen ions being pumped across the membrane. See, that hydrogen ion is going through this enormous protein thingy. Which means that these hydrogen ions are being pumped
against their gradient, and we end up with, on the inside of the thylakoid membrane here, we have a lot of hydrogen, and then on the outside there is not a lot of hydrogen. So this difference in the hydrogen gradient ends up being used later on to make ATP.
Photosystem 1! So we have our electron come in from Photosystem 2. Yeah, happy electrons. And it is going to get excited again by more sunlight. Yay, happy sun. And it's going to go up and bounce around across the pigment pads again, which are chlorophyll B.
And they're useful at different nanometer wavelengths or whatever, so it's more awesome sunlight types and stuff. And this electron, this highly energized electron, is going to be used to energize a high energy electron carrier called NADPH, which goes off to the Kelvin cycle. So this is an ATP synthase molecule, that big jellyfish thing right there. So remember that photosystem 2, that the purpose of it was to create a hydrogen ion gradient on the inside of the thylakoid. So that is the inside of the thylakoid, and all of those hydrogen ions are in there from photosystem 2. Now the purpose of this is to power that jellyfish. All of those hydrogen ions are going to want to leave the thylakoid because things want to go away from where all of the other same things are. If that makes any sense. If you don't know concentration rules, go look those up. So all of those hydrogen ions are going to go woo! They're going to go through that molecule, and that molecule is going to use that movement of those hydrogen ions to power the production of ATP. You should be clapping and freaking out right now because ATP is like the biggest deal ever. It powers everything. Everything in every cell needs ATP. So specifically, this ATP is going to be made for the Calvin cycle. And the Calvin cycle is quite silly and inefficient, but we'll see that it's very important for plants, and that they need all of this ATP to be used in the Calvin cycle. So yeah, that's what that is.
So sometimes we need to go through a process that is called cyclic light reactions, which go back through PS1 to make more ATP. This happens if there is not enough ATP to power the Calvin cycle, or if NAD plus is not abundant enough.
NADPH is not made in this version of the light reactions, only ATP. This is the dark reaction, which doesn't depend on light. It is also called the Calvin cycle. So this is the Calvin cycle. So this is where carbon dioxide comes in. The plants absorb it into their leaves through their stomata, which are tiny little pores in leaves. So 3 of those carbon dioxides come in, and the carbons from it, which are shown in pink up there, are going to get fixed or attached onto these little 5 carbon molecules called RUBP. And they come from the previous carbon cycle, which hasn't happened yet, of course. So that the carbon from the CO2 gets fixed onto the RUBP by rubisco, which is a super important enzyme and is actually the most common enzyme on the entire planet. Wouldn't you know? Super cool. So the carbon from the CO2 is shown in pink there again. And this takes 6 ATP to happen. And this first step is called carbon fixation. 6 ATP. 6 ATP.
So next we're going to go on to the next step, which is going to be where these 6 carbon chains are going to be reduced into 6 little molecules called G3P, which uses 6 NADPH to reduce them, which in turn regenerates NAD+, which goes off to PS1 to do its thing.
Then we're going to go on to the next step. Step 3, where our RUBP from the first step is regenerated. Because we have those 6 little G3Ps there, but we need to regenerate the 5 carbon molecules for the next Calvin cycle. So 5 of those G3Ps are going to stay and be regenerated by those 3 ATP, and one little G3P molecule is going to go off to be turned into glucose in the stroma. So the whole point of the Calvin cycle was to make that one little G3P to be gone off and made into glucose. And that's why I said that this is inefficient and silly, but it's not, because plants do it all the time, so I'm assuming it has a point. Alright, so it takes 6 rounds of the Calvin cycle to make 1 glucose.
So Leon asked us to include this lovely flowchart for your information. If we have no glucose, we have no plants, which means no O2 or food, which means no animals, which means no steak, which means you are dead, even if you do not eat steak. So, no plants means you are dead.
The end!
Thank goodness.